So last time we talked, I mentioned at the very end, after we talked about the model systems, that in many labs, people are using human cells, growing them in culture, and doing experiments on those. So before we start looking at the molecules that make up cells and how we obtain information and how cells function at the molecular level, I thought it would be important to give you a little bit of background on how these cells are grown, how they behave, and some of the practical aspects of using these cells. So we're going to talk about the properties of human cells in culture. And the first thing that needs to be done is to get tissue. Actually, this could be human or it could be mouse, but we're talking right now about human. You have to get that tissue and then take that tissue, break it up into single cell suspension, and then put it into tissue culture. So in a sense, this is a simple outline of how it's done. You take tissue, it could be a biopsy, liver biopsy, skin biopsy, any organ biopsy. If you're using mice, you can use mouse embryos. And back a long time ago in the 50s and 60s, when people are first starting to work this out, I'll talk about this in a little bit, people actually use human embryos, which is that's not done anymore, at least not very commonly. But in any case, you take the tissue, chunk of tissue, and you need to digest that or break it into single cells in some way. So we use a protease, which is an enzyme that chews up or degrades the proteins on the cell surface, which holds these tissues together so the cells kind of fall apart. Sometimes people push them through kind of a mesh that breaks them up into single cells. But in any case, once you get single cells, then you put them into a culture dish, and everybody's seen these either glass or plastic culture dishes. And into that culture dish, you put growth media. And the growth media is basically synthetic. You put in amino acids, which are needed for proteins. You put in all kinds of vitamins, all kinds of salts, sodium chloride, potassium chloride. You put in metals like iron, magnesium, and also you put in glucose. Glucose is used as an energy source, basically the food for the cells. Now, if you put all these things in, the cells won't grow. They'll only grow, most of the time, they'll only grow if you add 10% animal serum. Basically, in order to get the cells to grow, you have to add blood. It's not actually blood, it's serum. I'll describe that in a second. And that's 10% of the volume. And generally, the way it's been done over the years is the animal that's used is cows. And the way those are obtained is you go to, or there's industries that are connected with the cattle industry, which meat processing, and they drain the blood, and then they'll use that to prepare this animal serum. Sometimes horse serum is used. The serum is what's left after the blood clot. So if you take the total red blood, put it into a tube, and let that clot form, and you remove that clot, the yellowish material, that yellowish liquid is the serum. And in the serum are basically all kinds of growth factors, hormones, other proteins that are required to stimulate the cells to divide, other nutrients that may be micronutrients that we're not aware of. And so once you put these in with the rest of the media, then the cells begin to grow. Not all cells. That we still have a difficult time growing many types of cells. Some organ cells, like pancreas, is very hard to grow, and skin is actually very hard to grow. Uterus is very hard to grow. So we don't really know a lot about growing cells yet, but we're learning, and I'll show you in a little bit. We have some information on some specific cell types. But if we were talking about human cells and you took human tissue, what would happen is fibroblasts would begin to grow. It's kind of connective tissue, again, in the presence of this animal serum, which is kind of a black box. We put those cells into culture, and normal cells, as I have written on the bottom, normal cells may grow, depends on the tissue, but they do not grow indefinitely. They only grow for a very short period, and this is a very important point, which we'll talk about in the next slide. So these are results that you get when one puts in normal human cells in culture. You can take normal liver, or you can take normal connective tissue, excuse me, and you'll get cells to grow. This experiment, an experiment very much like this, was done in the early 1960s by an individual called Hayflick. And he was interested in basically getting cells to grow, and he did this experiment, which I'll describe right now. What he did was he took human embryos, and I told you at that time, in the 60s, people were able to use human embryos. It's not done so commonly now, but he took those human embryos, and he was able to get single-cell suspension, and he put them into tissue culture with the serum. And the cells grew. So what you have, the way I have this described is on the x-axis, it's the number of cell generations. That's basically the number of cell divisions. So you have one cell division, there's 25 cell divisions, and 50 cell divisions. And then on the y-axis, it's relative growth. That is basically how fast these cells are dividing. Now there's two lines there. One represents embryo cells, and that's kind of on the outside. What he found was that when he put these cells in culture, they began to grow. And in fact, they grew quite well. They increased their growth rate for a period, and then they kind of stabilized. They stabilized for quite some time. And that stabilized cell growth is referred to as a cell strain. And then they went through what we call a crisis. That is, every time he did the experiment at around 50 cell generations, the cells stopped dying. And the culture crashed. They went into senescence, they died, they were not able to be grown past 50 cell generations. He repeated this many, many times. Let me just give you some definitions. So primary cells, which are on the very left-hand side, the primary cells are the cells that are taken directly out of the tissue, put into culture for 1 or 2 or 3 generations, cell divisions, and these are thought to represent most accurately the cells that are in vivo. So a primary culture represents as best as we can cells that exist in the body. The cell strain are the cells that are growing for those 50 cell generations. And then, of course, that crisis is when they crash, they stop dividing, and they die. So no matter how many times he did this experiment, he found that the cells would begin to die at around 50 cell generations. So he hypothesized at that time that there was some kind of a internal clock, internal counting mechanism that allowed these cells to keep track of the number of cell divisions. They appear to have some kind of internal clock mechanism. And he presented that in 1963. I have that written down towards the bottom. He tested that by taking adult cells. So here, the first example was using embryo cells, but when he took adult cells, these were adult biopsies from lung tissue, and he put them into culture, he saw something very similar. The primary cells, they grew for a while, increased their growth rate for some time. They grew as a cell strain, but routinely they would go into this crisis much sooner, anywhere between 25 and maybe 35 cell generations, and the cells would go through the crisis and die. So what he proposed was that there was some kind of counting mechanism, some internal clock in a way, that counted the number of cell divisions. 50 cell divisions was kind of the maximum that you would get out of an embryo, and then adult cells, presumably since they had already gone through a series of cell divisions, that they had already expended some of their growth potential, and so the Hayflick limit was reached much earlier. So we believe there is a counting mechanism. There is some kind of a clock that counts the number of cell divisions in a sense, and we now think we understand what that is, and those are the role of telomeres, one of the role of telomeres, which are structures that are at the ends of chromosomes. And the telomeres are thought, and there's evidence to show this when we talk about DNA replication and telomeres. I'll talk about that later. In 2010, so 50 years after he made this discovery, it was determined that there is in fact a counting mechanism, and that counting mechanism is carried out by telomeres. So there is a clock. The Hayflick limit is real. Now, those are normal cells. Normal cells have a limited lifespan in culture. They cannot grow past 50 cell generations. So it makes it a little bit difficult to use these normal cells for experiments if you need them to grow for a long time. They stop growing, so there's only a limited amount of time you can use these, but they're often used. And it turns out that there are two types of cells that we know of that are considered immortal. That means they can grow indefinitely in tissue culture. They ignore the Hayflick limit. And so the only cells that can grow indefinitely in culture are cancer cells, which are defective in many ways. One of the problems with cancer cells in the body is that they're growing without control, and that seems to carry over into tissue culture. And the way we phrase it is they bypass the Hayflick limit in some way. And there is an issue with telomeres. And also stem cells. Stem cells are also immortal. Stem cells are premature cells, or immature cells. I'll talk about those in a few minutes. And the way people think about it is they're so premature in a sense that they don't activate the Hayflick limit. So in any case, cancer cells and stem cells are immortal. Now, cancer cells are the cells that are used extremely frequently in research. They can grow forever in culture, so it makes them very convenient. And the classic example is HeLa, a cell line called HeLa. HeLa cells are grown all over the world. Every lab grows HeLa cells. They've been growing in culture since 1953, continuously in different labs. We have them in our lab. Just about every lab has HeLa cells that they grow. And so they've been growing for quite a long time. They originated from a woman with cervical cancer. Her name was Henrietta Lacks, which I want to talk about because I think it's very timely in a sense politically to talk about this story, what happened with her. So HeLa cells are widely used. They've been grown in culture since 1953. Let me just give you a little background on the origin of these cells. So HeLa cells, again, were first put into culture. Here it says 1951, so that's accurate. They're immortalized. They grow forever. And HeLa cells are classic. There's a story behind the HeLa cells, which is quite unfortunate. And that is they were taken from a woman, Henrietta Lacks. She was a young 31-year-old black woman, was in the hospital being treated for cancer, cervical cancer. And basically she was used, in a sense, to generate this cell line. Her tissue was taken, her cells were taken without her knowledge. She had no idea what was going on. And they took those cells and they were used without compensation. She never knew, and she passed away from this very aggressive cancer. There's a picture of her on the left with her husband. They had children. Not only did she not know that these cells were taken, and not only was she not compensated, but these cells became critical in molecular research. They were used by dozens of pharmaceutical companies to generate drugs, to generate vaccines, to study the human genome. People got famous using these cells. People made millions, and pharmaceutical companies made millions and millions and millions of dollars using these cells. And no one never knew the origin of them. It was kept a secret. Her family never knew. Until a woman came along, Rebecca Skloot, who was very interested. She was a journalist. And she heard about this story, and she heard some rumors, and she became very interested in the origin of these cells. And basically, she did her research. It took her quite a number of years, and she discovered who it was, Henrietta Lacks. Contacted the family. The family never knew. They were all very, very poor. No health insurance. Living in poverty, and never knowing that her mother's cells were used to make so much money. So it became a big issue. As a result of that, there's been lots of changes in the medical system, and that is there has to be informed consent. Anytime you go in for surgery, you have to sign papers. If you look carefully at those papers, they may say that they may take some of the tissue for research. She was not, she had no idea that this was going on. That you sign the papers, called informed consent, you sign the papers that they can use the tissues for research, and also in small print that you will never get any compensation for it. Most people sign it, but you don't have to sign it. So the immortal life of Henrietta Lacks. Also, it became clear that since the HeLa cells were sequenced, the DNA genome was sequenced, it was public, that also became an issue, because what that meant was that the genetic information of all of her offspring also was public, and again, without their knowledge. They hired lawyers, people to represent them, and I believe that it's very difficult to get the sequence of HeLa cells now. I think you have to get special permission, and you can get it, but because we want to try and protect the integrity of these individuals who are her kids and grandkids. So it was a very, in a sense, sad story. A black woman who was kind of taken advantage of. There's this book that came out, Immortal Life of Henrietta Lacks. I think there's also a play that came out. Some people may have seen it. And one of the readings we're going to take a part of this book and read that this semester. Take a look at that. So that's the story of the origin of HeLa cells and Henrietta Lacks and her family. Let's just take a look, move on, take a look what happens not just in human cells, but also in mouse cells, because that's also important. So what happens in mouse cells is analogous to what happens in human cells. So you take mouse tissue, you can take mouse embryos, put them into culture with the media, with the serum. Again, I have on the x-axis number of generations, then the relative cell growth on the y-axis, and you can take these mouse cells, put them into culture, and they look like they behave very similarly to human cells. That is, you have the primary cells at the very beginning. They grow for a while. They develop the cell strain. And then at an early time, not 50 generations, but somewhere around 25 generations, they go through a crisis and they stop growing. But they don't die. They sit on the dish and they divide. But they divide extremely slowly. They may divide every 6 days, every 2 weeks. But they're alive, but they're just growing extremely slowly. And then at one point, which is called the transition point, at one point, a subpopulation in that culture starts to divide and starts to grow. Something changed, something happened. And then the cell growth picks up, and you can see it increases, and it becomes immortalized, and becomes what we call a cell line. So cell strain, either in human or mouse, is a temporary growth of the cells, and the cell line is more permanent. So all the cancer cells, HeLa cells, are cell line. And these immortal mouse lines are grown in culture for long periods, and so people use those for research. Again, they're not normal. They become immortalized. People have actually studied these to try and sort out what would cause these cells to grow, look at mutations or look at the defects in the genome. And in fact, some of these cell lines can cause tumors if you inject them into mice. So something happened during that transition point that makes them tumorigenic, and so some scientists are looking at what are some of those changes, because maybe it duplicates, in a sense, cancer formation, but in the dish. So there's a number of cell lines that are used. I have those written at the bottom. They usually are named after the strain of mouse. So NIH is a mouse, a type of mouse. NIH mice, it's got NIH3T3, that's the cell line. BALB is another mouse strain. BALB3T6 is another cell line. Swiss is another mouse strain. Swiss 3T3. There's some that are been derived from rat. There's one called rat1. So there's dozens and dozens and dozens of these different mouse cell lines. In the literature, a lot of times you'll hear people say they're normal cells because they came from normal tissue or they came from an embryo, but they're nowhere near normal. They've undergone mutations, they've undergone gene rearrangements, translocations, all kinds of changes that people are trying to sort out, and in fact some of them can make tumors, so they're clearly not normal. So that's the generations of the cell, the types, the way we generate cells to grow. Now, what I had told you so far is that these are in the presence of the serum, but as time goes on and people are studying different kinds of cells, we are starting to learn what kinds of factors, rather than just simply adding 10% serum, what kinds of growth factors or what kinds of hormones are required to grow cells. So this is shown in the next table. So here's a table showing a number of different more specific cell types that can be grown in culture. Turns out all these are neural related cells, but other cells can be grown too. And what you see in this particular table is you have one column shows the differentiated cell types, so cortical neurons, and you have astrocytes, and you have retinal cells. And then on the right-hand side, it shows the relevant medium. So DMEM F12, for example, is a commercially available media that carries all the vitamins and amino acids and sugars and so on, the basic salts and so on that are needed. But the cells cannot grow unless you add either serum, like I mentioned, 10% serum, or in this case, people have worked very hard at trying to sort out what specific factors or hormones are required to get the cells to grow. So this is kind of an ongoing area of research, and that is learning how to grow different types of cells. So for example, I'll just we'll look at a couple of these. So the first, up at the top, you have ventricular zone radial glial cells. It says relevant growth factor NA. I think that means that it's not applicable, and that's because people have not figured it out yet. So they're still using 10% serum. But if you look at the cortical progenitor cells and the cortical neurons, for example, here we are slowly starting to be able to sort out which factors are required. So here they have relevant growth factors. And the BDNF stands for brain-derived neurotropic factor, brain-derived growth factor. GDNF stands for a glial-derived neural factor. So these are specific proteins that trigger the cells to divide. NT3 is another one. Laminin, and then beta FGF is beta fibroblast growth factor. So with specific proteins, specific factors, we can get these cells to grow. So little by little, cell culture is getting much more sophisticated. Now, all these cells are grown in culture on a dish. They do not represent tissue. You can study the cells, you can study the behavior of the cells, but you cannot really study how they function as a tissue, as a coordinated tissue. So there's a lot of research going into the development of what are called organoid cultures. And we have a few groups here in our department, also in the pharmacy department, that are working on developing organoid cultures, because organoid cultures are cultures that may more clearly represent tissue, specific tissue. I'll give you an example here in the next one. So here you have the development of organoid cultures. And basically they're just, it's very simply showing you they take a biopsy, they break the cells up, like I mentioned with the digestion, they put them into culture, but the culture is different. It's called a 3D culture. The cells, rather than sitting on the bottom of the dish, are sitting in kind of a gel-like medium. They call it a semi-solid medium. It's a gel-like medium where the cells are not sitting on the dish, but they're suspended in this culture with the proper media. And what happens in some cases is that they start to aggregate together as they divide and then grow into something that starts to look maybe like the tissue. So there's been a few different kinds that have been worked on to get these 3D organ cultures. There's stomach, intestine, liver, prostate, pancreas, and colon. And they form 3D cultures. They start producing the enzymes that are, let's say, produced in the intestine. They start to take on some of the shapes that intestine or colon will take on. And these are becoming extremely important, these organoid culture, extremely important for studying, for example, let's say we're just talking about cancer, chemotherapy, that before we even go into a mouse, that one can study treatments rather than using cells, but using these small organs to ask whether or not this particular treatment will have an effect on how the organ functions, or other drugs that may be used to, let's say, increase the function of the organ. It doesn't have to be cancer. Let's say inflammatory bowel syndrome, using drugs that might be able to correct that, actually using, in this case, organ cultures. Okay, so the bottom line here is that, and again, these don't grow indefinitely in culture because they're normal. So they'll grow for some period, and then you have to start new ones. So the bottom line, everything I've told you so far, is that normal human cells have a limited lifespan in culture, Hayflick limit, about 50 cell generations. Mouse cells are a little bit different. They can go through this transition phase and then start growing into a cell line, which then are immortal. Cancer cells are immortalized. They can grow forever and ever in culture, like the HeLa cells. And also stem cells, I had mentioned earlier, grow indefinitely in culture. So I want to talk a little bit about stem cells. So let's talk a little bit about stem cells. So stem cells are required for what we call tissue renewal. And as I wrote on the first line there, stem cells generate a continuous supply of what we call terminally differentiated cells. Terminally differentiated cells are the cells that carry out the function, like terminally differentiated skin cells, or colon cells, or brain cells. So let me show you an illustration in the next slide. So here's a kind of a perfect example of what a stem cell is. So you have a stem cell which undergoes cell division, and as the cells divide, it's asymmetric in a sense. One cell, shows on the left, one cell goes back, remains a stem cell. But the other cell of the pair goes on to differentiate, and that's shown in red, goes on to differentiate into more mature cell types. So it shows a proliferating precursor cell, and then finally terminal differentiation. And this I'll show you, can be colon, it can be any organ type. Any organ Every part of our body has stem cells to replace cells that are lost as they differentiate, as cells slough off, as they die. So here's an example, pretty classic example that takes place in the blood. So here we have what we call self-renewal of a hematopoietic stem cell. The hematopoietic stem cells are among the class of tissue-specific stem cells. So you have stem cells in the blood, you have stem cells in the gut, you have stem cells in the skin, stem cells in the liver, stem cells in just about every organ, bone stem cells that can continue to divide and give rise to mature bone. But they're tissue-specific. So here, if we look at the hematopoietic stem cell, what it's showing is that in the bone marrow, these occur in the bone marrow, that you have these stem cells that give rise to every single type of cell in the blood, the T lymphocyte, B lymphocyte, which produces the antibodies, neutrophils, monocytes, red blood cells. All these cells are derived from a self-renewal hematopoietic stem cell. I want to just stop for a second and just tell you a little bit how that impacts something like chemotherapy. Most of the chemotherapies that are used today are not really super specific. What it is they kill all rapidly dividing cells. Cancer cells are rapidly dividing, and so the drugs enter the cell and they damage DNA, and they kill or allow the rapidly dividing cells to be killed, but they don't have any target specificity. So any cell that's dividing will be killed by the chemotherapy. And that's precisely why, if you've ever had chemotherapy, or if you know anybody who's had chemotherapy, that is, what happens is they have a lot of problems with the levels of these cells in the blood. So people who are on chemotherapy get anemic. That means their red blood cells drop. Their B lymphocytes and T lymphocytes drop. Their immune system drops, so they're not producing sufficient antibodies. The neutrophils drop, the monocytes drop, all these cells drop, and particularly the megakaryocytes, which give rise to platelets, and platelets are involved in blood clotting. So if you have had chemo, if you know people who have had chemo, they are always going in to get their blood checked, and invariably the numbers drop, and they have to wait till they come back up in order to get the second round, another round of chemotherapy, or in some cases they'll actually get transfusions. So the reason why you have these problems is because the chemotherapy is not specific. Here's another example, and this is the colon. So this shows kind of a section of the colon. The inside of the colon has what are called villi, which are kind of they're like hairs. Or they're not hairs, but they're columns of cells that grow from the surface. They increase the surface area of the colon in order to digest food better, and they're constantly being regenerated, because as we digest and as we eat and as things go through, these things get sloughed off, and we have to continually regenerate them. They're regenerated at the bottom of what's called the crypt. So if you look at the top, it's the lumen of the gut. The villus shows those large increases in the cells, these hair-like structures that move the food around. And at the bottom of each villus, there's what's called the crypt. You can see that labeled on the left-hand side. And the crypt is what gives rise to the mature cells. So as these things get sloughed off, they have to regenerate. And they regenerate, if you look at the bottom of the crypt, you have the yellow cells, which are, well, you have let's say with the blue cells, they're non-dividing cells, they're terminally differentiated. Those are rising up into the top. The yellow cells are dividing cells in order to generate the material, but at the very bottom, you see the dividing stem cells in red. And those dividing stem cells are required to generate all the cells that will give rise to these villi in order for the colon and the gut to work properly. And again, with respect to the chemotherapy, quite often people have pretty serious digestive problems, and that's because the stem cells are also eliminated. And so people are not developing the villi in order to make the tissue work properly, efficiently. It's also why people lose hair, because in the bottom of the hair follicle, there are stem cells that are constantly reproducing the hair structure, producing the proteins that make the hair. So when those stem cells are lost, then people lose their hair. So all these things are killing dividing cells. So these are tissue-specific stem cells. So that's what I have written in the second line, tissue-specific stem cells versus embryonic cells. So we just saw the tissue-specific, for example, give rise to the blood, give rise to the cells of the colon, they give rise also to the cells of the skin, versus embryonic cells. And there's been a lot of interest in embryonic cells because embryonic cells are what are called totipotent or multipotent. They can give rise, because they're so immature, they can give rise to every tissue and every organ in the body. Now, it's been an issue because working with embryos is not very easy to do. You have to get special permission from the NIH, National Institutes of Health. It's very difficult to work with embryonic tissues, human embryonic tissues. People have been doing a lot of work with mouse embryos. But there has been some work using human embryonic cells. Let me just take a look and show you how this is done. So embryonic stem cells are derived from early 5-day embryos. So this would represent either a mouse or a human embryo. In human, it would be 5 days after fertilization. And this is, what you see on the left is referred to as an early embryo or a blastocyst. A blastocyst contains approximately around 1,000 cells. And there's two cell types. There's what's called the trophoblast. Those are those gray cells on the outside. And the trophoblast will give rise to extra embryonic tissue, like the amniotic sac or parts of the placenta. The cells of the inner cell mass will ultimately give rise to the entire embryo. So these cells, there's only around 100 of them at this point, these cells are considered totipotent. That is, they have the ability to differentiate into every single cell type in the body. In fact, there's been experiments that have been done with mice where you can take these cells of the inner cell mass and get them to redevelop into an early embryo and develop into a full, complete mouse. So that these cells of the inner mass can develop every single organ and tissue, including the entire organism. And in fact, I'll say even one more thing. There's been experiments done with cattle where if you take cells of the inner cell mass from a cow or cattle, that these can, if they're handled properly, can develop into an entire cow. So you get the cells of the inner cell mass, you get the early embryo growing, and then you transplant that embryo into the uterus, and then you end up developing the entire cow. So there are completely cloned herds of cows. Each cow is identical because it's coming from the same embryo. So these are cloned organisms, so you have cloned herds of cattle. But for our purposes, you can take these cells of inner cell mass, put them into culture. It's called cultured ES cells, embryonic stem cells, with very specific media. And then, if they're treated in the right way with the right factors, the right hormones, the right growth factors, they can differentiate into any cell type that we would want. So a fat cell, neuron, macrophage, as it says, heart muscle cell, nerve cells. So that's what the power of these things are. People are trying to study what are the signals that would allow these embryonic stem cells, let's say, to develop into heart muscle cells or neural cells. And we hear about that because it can be used potentially as treatments for, let's say, spinal cord damage. If you damage spinal cord, those cells are not replaced. But if you can introduce embryonic stem cells that would differentiate into a neuron, then you may be able to repair that damage. Same is true with heart. If you have a damaged heart attack, that there may be ways to introduce these cells into the heart and get them to differentiate into the proper tissue. So there's been a lot of work looking at how to differentiate these things into these different tissue types to be used for therapeutics. It has been used successfully in some cases. There's been a lot of work with glial cells, and there's a form of blindness, it's called macular degeneration, where some scientists and physicians have been able to recover the sight by introducing embryonic stem cells into the eye. The idea is that although we may not know what factors or what hormones or what growth factors may be required to induce these different cell types, that if you place the cells into the proper environment in the body, then those factors will be there and cause those cells to differentiate into the right tissue type or cell type, and that seems to be what's happening. So if you introduce cultured stem cells into the eye, they will differentiate into glial cells. If you introduce into the spinal column, it looks like they will also induce into neurons. So there is hope there, and people are, of course, trying to work out the conditions so it can be used better for therapeutics. Now, since embryonic, particularly human embryonic stem cells, have been very difficult to work with, mainly because it's not legal if you are funded by federal funding to work with embryonic human embryos, people have been trying to get around that by using what are called induced stem cells. Let me just show you those in the next slide. So here we're talking about what are called induced pluripotent stem cells, IPS cells, and they're often, it says, often used to replace embryonic stem cells because the embryonic stem cells are kind of hard to come by. And so what's been discovered is that there's 3 different genes that can be used to introduce into a mature cell like a fibroblast and get that fibroblast to revert back into a stem cell. It's a little bit bizarre. You're taking a mature cell and causing it to go back in time. So there's 3 genes. One's called OCT4, one's called SOX2, another's called KLF4, and people have done this a number of different ways. Introduce those genes into the nuclei of the fibroblasts, and then those fibroblasts go back in time and revert into a stem cell. And then those stem cells can then be used to differentiate in culture, like we were talking about earlier, smooth muscle cell, neuron, fat cell, and so on. So the IPS cells are now used to learn what factors can be used to differentiate these things into these different cell types. Now, nobody is using these for any kind of therapeutics. The reason is because a number of these genes, the OCT4, SOX2, are genes that have been related to cancer. So there is some likelihood that some of these induced pluripotent stem cells may be tumorigenic. So while nobody would ever introduce these cells into a person, they are being used to differentiate in culture and trying to learn more about it. So that's a way to get around using stem cells, the embryonic stem cells, but of course they can't be used because they could be tumorigenic. Okay, so I want to just end this discussion on cells by talking about what's called the cell cycle, and just kind of review that for some of you who may have had this before. For some who haven't, we'll look at it for the first time. I'm not going to talk a lot about it, just to give you a sense of how these cells undergo cell division. So this is referred to as the cell cycle. Drawn as a cycle because if you have cells that continue to divide, it keeps going through these different phases. So let me describe what we have here. So we have a number of different phases. Let's start with G1 phase. So G1 phase is a phase where cells are preparing for cell division, for DNA synthesis. That's in blue. S phase is shown in the center in orange. S phase is DNA replication. G phase is another phase of the cell cycle, where cells are preparing for mitosis. And then we have M phase, which is basically mitosis, which takes a pretty short time, but this is where you have the condensation of the chromosomes into the mitotic chromosomes, and then the splitting, and then you undergo what's called cytokinesis or cell division. Now, if a normal cell division cycle is around, let's say, 22 to 24 hours, the large bulk of that is what's called interphase. Interphase is the time of the cell cycle where you can't see anything happening. So it's called interphase. Interphase is the phase that takes place between cell divisions, between mitosis. Now, in my opinion, there's a little bit of it's like a little bit of a misnomer. You say interphase, it sounds like nothing's happening. But in truth, there's an enormous amount happening during interphase. Let me just break it down. During the G1 phase, so if you start at the top of the G1, say the cells are resting and they're stimulated to divide by some kind of a growth factor. It's a hormone that would stimulate the cells to enter G1 phase. So G1 phase takes 10 hours. You can't see anything happening, but during that 10-hour period, the cells are producing all of the enzymes, the DNA polymerases, all the histones, all the enzymes that are required, all the proteins that are required for DNA replication. There's an enormous amount of activity happening during G1, and it takes a long time to prepare what needs to be prepared. So it's not that nothing is happening. There's a lot happening. The cell is changing and producing everything it needs for S phase. S phase, where also you can't really see anything if you're looking at the cell, so you call it interphase, as if nothing's happening. During S phase is when DNA replication takes place. And this, of course, is extremely complicated, a lot going on. The DNA is doubling, and later we'll talk about the mechanism of DNA replication. And this phase takes approximately 8 hours. Then we have G2 phase, which again, you can't see anything happening. So you don't know, so they call it interphase. It's like nothing's going on, so it's like a resting state. But anything but resting, during the G2 phase, it is producing all of the proteins that are required in order to undergo M phase. M phase only takes about 2 hours. And of course, during M phase is when you have the chromosomes condensing into the mitotic chromosomes that we're used to seeing, the splitting of the mitotic chromosomes at the centromere, the movement of the chromosomes, and then the splitting of the cell into two. So that we can see. That you can actually see happening under a microscope. So when people think about cell division, they say, oh, there's interphase, like a resting state or whatever, and then M phase is where we look at what's happening, and it's like, oh, there's a lot of activity there. But the fact of the matter is, and that's kind of what I want to stress, there's a lot of activity happening in all these various phases. Now there's other additional activities that I want to superimpose on that. And these are called checkpoints. And we'll look at that in the next slide. So here again we have the cell cycle with the G1 phase, the S phase, the G2 phase, and the M phase mitosis. And what I want to stress here is that in addition to these different phases producing the components they need to carry out the next phase, like G1 is preparing what it needs for DNA synthesis, and G2 is preparing what it needs for mitosis, there's other activities that are going on, and those activities are monitoring the status of the cell. So there's what's called a G1 checkpoint. The G1 checkpoint, what it says here is the environment favorable? What that means is really, are there any mutations in the DNA? Are there any defects in the DNA? Because if there are defects in the DNA, or if there's mutations in the DNA, the cell has a way to identify that and then not undergo DNA synthesis. Because the idea is you don't want to replicate damaged DNA. So the G1 checkpoint is identifying lesions in DNA. Now what it does is, if the checkpoint is activated, it stops cell division and does one of two things. It either allows the cells to allows the cells to repair the DNA damage, or if the DNA damage is too severe, it'll actually allow the cells to die, which is a process called apoptosis. So the cells will enter apoptosis and eliminate the cell. The idea is rather eliminate a cell with a lot of damage than undergo DNA synthesis and replicate that damage. Now there's another checkpoint up in G2. There's a G2 checkpoint. And that is a little bit more descriptive. What it says in the blue highlight is all the DNA replicated and is all the DNA damage repaired. The idea again is that we don't want to enter mitosis. Can't start entering mitosis if the DNA is not replicated. Because if the DNA is not replicated and we start to enter mitosis, the cells basically get screwed up. Or is the damage repaired? If the damage again is not repaired, then again we don't want to enter mitosis and generate cells with damaged DNA. So again, it will stop the cell division, and generally what happens in this case is apoptosis. Cells will basically be killed. And then finally there's an M phase checkpoint. And what happens during M phase checkpoint, and that is prior to the actual cell division, and it's asking the question you can see on top, are all the chromosomes properly attached to the mitotic spindle? They have to be attached to the mitotic spindle in order to be pulled apart. If they're not attached, then when cell division takes place, you get more than one copy in each cell and no copies in another cell. So these are extremely important checkpoints in order to evaluate the status of the cell, because the chromosomes are going to be pulled apart, and if they're not pulled apart properly, then you don't want to divide. And so what will happen here is the cells will undergo apoptosis. So the last thing I want to say, and then we'll close here, and that is defects in these checkpoints, the S phase checkpoint, the M phase checkpoint, and the G2 checkpoint. These are defects. There are defects in these checkpoints, and these are all usually, or I would say always, associated with cancer. So we know that cancer is due to mutations in various genes. And a lot of the genes that have been identified which give rise to cancer are defects in genes that are responsible for these checkpoints. And so, for example, if you have a defect in the S-phase checkpoint, what happens is, rather than these cells being repaired, or rather than these cells undergoing apoptosis and eliminating them, you're replicating DNA with damage, introducing more mutations. Or if you have a defect in the M-phase checkpoint, where you need to do that in order to have the chromosomes pulled apart properly. If there's defects there, then again, rather than undergoing apoptosis and eliminating those severely damaged cells, the chromosomes get screwed up, and you get different cell numbers and you get different chromosome numbers with different cells, and the cells become genetically unstable. So the apoptosis in these checkpoints are crucial. Okay, we will stop here, and next time we will start talking about molecules.